In this video we're going to show you how to load samples onto an SDS page gel and run an electrophoresis. The first step is to add the electrophoresis buffer into the inner chamber where our gel is. We fill it up to the top, and then we can take out the comb if you haven't done that already. Just pull straight up like this, and then you have the wells formed where we will load our samples. So once we do that, we wait for a couple of minutes to make sure that the inner chamber is holding the buffer properly and it's not leaking. If it's leaking, we can fix the setup and do it again. So just take a moment to make sure that it's not leaking, and it's not. So now we can take the electrophoresis buffer, and I usually load it from the inside. So this acts to wash out the wells a little bit, and you just have to add enough buffer to complete the electric circuit. So you have to get the bottom of the gel also covered about halfway up the doors, and this is sufficient to complete the electric circuit and carry out the electrophoresis. So now once we have the buffer set up, the gel set up, with the buffer added, we are ready to load our samples. So in this case, we have 4 different samples that we're going to load that we see here. So the first sample is an Eppendorf that has a molecular weight ruler. So these are pre-stained proteins. It's called page ruler in this case. It's a series of pre-stained proteins that will tell us the relative molecular weight of our proteins of interest. Okay, so it's a standard. Then we have our 2 samples of our problem proteins. So this first one is a crude extract that we're going to do in this example, and then in the second one we have a purified protein. So this is a protein purified by affinity chromatography in this case, but these could be any samples, right? And then the last one in our case is going to be a standard of BSA, and we're using this in this case as an internal standard for protein amount. So we have the molecular weight standard and the BSA standard, which is going to show us what 10 micrograms of protein looks like on one of these SDS page gels with our staining system. So again, these samples could be of any origin or type. You just have to make sure that they are resuspended in Lemley sample buffer. And it's interesting sometimes to use these types of internal standards, although they're not necessary. Almost all gels will have the molecular weight marker on one end usually, but again, you can separate any type of protein samples on these polyacrylamide gels, SDS page. So we're going to load the gel. I'm going to show you how to load the gel. So the first lane will be our molecular weight marker, and we're going to load 5 microliters in this case. Depends on the preparation, but this one with 5 microliters we can see it very well. And the best way to do this is to angle your pipette towards you, right? At this angle, not straight up and down, at an angle towards you. And then what we do is insert the tip of the pipette into the chamber where the electrophoresis buffer is. We touch the large plate and then slide down the glass plate until we get to the small plate. Okay, you can see here how you just slide down, and you can see how it just stops. The pipette will stop when it gets to the small plate. So then for sure you're between the 2 plates, and then you just have to move yourself horizontally so you're on top of one of the wells, and then just press down your pipette and dispense your sample into the well. You want to go slowly and steadily. You don't want to do it too fast because you don't want it to basically spill out of your well. Okay, so now we're going to load the second sample. So these samples are made with usually diluted with SDS page buffer, so Lemley sample buffer in most cases. It has bromphenol blue and also something to make it more dense, so it will fall to the bottom of your well. So it's the other thing that this Lemley sample buffer has is SDS. So this is a detergent, so it's very easy to make bubbles, and this is what we have to be careful to avoid here. So what you don't want to do is to be pipetting up and down your sample. You just want to take out your necessary volume and then apply it to the gel in the fashion that we just showed you before. Okay. So in this case, we're going to load all of the volume of this sample. And again, here it's very important not to pipette up and down because you'll create a lot of bubbles. So just take our pipette, put a tip on, set the volume that we need, and just pipette up the volume that we need. And like I said, insert it into the volume, into your sample, and take up the volume that you need, and then just stop pipetting, right? Don't pipette up and down, and don't let a lot of air bubbles get into your pipette tip. So once you have it like that, again, the angle towards us, insert into the buffer, touching the small plate and on top of the well, and then just dispense your sample. And like we said, carefully and slowly. So our next sample in this case has a higher volume, but this would depend on your experiment, of course. So in our case, you can see the sample is yellow because of there's a decrease in pH. This is more acidic, this sample. So the bromophenol blue is a pH indicator. And in this case, we want to load 30 microliters of our second sample. So again, just very, very carefully insert your tip into the volume and just take up your sample without pipetting up and down. And once you arrive to the end of the sample, just stop pipetting. And at the angle, touch the large plate, go down until you get to the small plate, and then just dispense your sample. And you can see here, once we touch the electrophoresis buffer that has a high alkaline pH, our sample turns again to the blue color, showing again the bromophenol blue pH indication. Okay, so in the last case, we're going to add our BSA standard in our example here for today, 20 microliters. And the same thing, this is also in Lemley sample buffer. So we want to make sure that we're not pipetting up and down and creating a lot of bubbles. So we take up our 20 microliters in this case. So you can see here, just pipette and without creating bubbles. And again, the angle, as we said before, is towards you. This is very important. So this is the angle that you want to use. And this helps to make sure that you're between the two plates. If you do it straight up and down, it's easy to make a mistake. And if you do it this way, you can touch the large plate and just slide down until you get to the small plate and get on top of your well, and then just dispense your protein sample into the well, and then we can separate it by applying the electric current. And so the proteins will run down to the bottom of the gel and be separated on the basis of their size principally. So here's the cover we're going to put on now. Remember, the electrodes are color coded, red and black. So make sure that you match the color. So the red part of the cover goes with the red electrode, and the black with the black. Okay. So just place the cover on, push it down, and then we have to connect the wires to the power supply. And again, the power supply outlets are also color coded, red to the red and the black to the black. And the red is the positive current, and the black is negative. And then we're going to turn on our power supply, and we're going to set it to about 110 volts. In this case, it's going to be 109 constant voltage. And in this case, it takes about 1.5 to 2 hours to completely run the electrophoresis. So we will leave that running, and that's how you do an SDS page gel electrophoresis.